All right, so for chapter 10, we're going to look at maybe controlling microbial growth in the body. And so in chapter 9, we looked at controlling microbes in the environment, thinking specifically of surgical tools, surfaces. We talked a bit about air and removing something from liquids. But in this case, in chapter 10, we're really focusing on things that can go into the body and treat microbial growth more from a perspective of from the inside. And so it's much more limited what kind of chemicals can be used for that, because very harsh kinds of chemicals are not going to be able to be used inside the body. And we're going to talk in a lot of general terms, and then we'll focus on a few specific drugs as well. So, but there will be a lot of drugs that are mentioned, and then I will give you a short list of particular drugs you definitely need to know, but a lot of general terms and definitions also that have to be learned. All right. Okay, so if we use the term drug, we are describing a chemical that affects physiology. Physiology is the function of, in our case, the human body, and it affects it in any manner. So a drug can be legal or illegal. That is a decision of the government. But any drug, if we use the term drug, it's a chemical that affects physiology. And then really what we are talking about, we need to narrow in for our course. We will be talking about what are generally referred to as chemotherapeutic agents or chemotherapy. I know that when you hear the word chemotherapy, you probably think that that term is restricted to a cancer treatment. But in fact, that term just means what it sounds like. It's a chemical therapy. It's a drug therapy for any kind of disease. So when we're learning this, we're going to use a very broad definition of chemotherapy, and we're defining it as a drug that works against a disease. But disease can be caused by things that you inherit, so a hereditary disease. Diseases can be caused by something you encounter in the environment. You could get lead poisoning, for example, and then also infectious or microbial diseases. And of course, in this class, we're focusing only on microbial diseases. Okay, so we're not going to talk about chemotherapies that are used for hereditary type disorders. We're not going to talk about chemotherapies for something you get only from the environment. We're going to focus in on microbial diseases. And so that's why I highlighted at this bottom part. Antimicrobial agents is going to be the focus here. Drugs that treat diseases that are caused by microbes. On this slide, I have a lot of names. The name that you really need to remember is Alexander Fleming. But he wasn't the only guy. There was a guy in 1902, and this is actually a short list even, but Paul Ehrlich came up with the idea that there could be chemicals, chemotherapies that could kill a pathogen without killing the patient. And they started using arsenic to treat syphilis. Syphilis was a very common disease in these days, and they didn't exactly know how it was passed on, and but they used arsenic, which is a poison, but they could kill the syphilis without quite killing the patient. Then they would consider it effective. Nowadays, not the top choice. But by 1928, we have kind of a what you could call just a lucky discovery by Alexander Fleming. He was studying bacteria, and on one of his agar plates, there was a spore of a fungus that grew on the agar plate. And we've seen that. You accidentally can get from the air these fungus spores that can grow, and they make these like pretty large, fuzzy kinds of colonies. And what he noticed was right around where that fungus was growing, none of his bacteria was growing. And we call that in modern day, we call that the zone of inhibition. But he noticed that something being released from that fungus was preventing the growth of the bacteria that he was actually trying to study. And so we call this just a lucky discovery, serendipity, where he discovered penicillin, which is what we ended up calling the drug, that's made naturally by a fungus that has a genus classification, Penicillium. So the Penicillium fungus releases a chemical that we call penicillin, and it prevents bacteria from growing. So really, Alexander Fleming, quite by accident, discovered one of the first naturally synthesized by a fungus drugs that could then be used to treat microbial diseases. And you kind of scan down Gerhard Domack and Selman Waxman. They did some work. Oops, let me move my picture around here. Gerhard Domack found a drug called sulfanilamide, which is used to treat bacterial infections. And Selman Waxman discovered antibiotics from soil-dwelling bacteria. So again, naturally occurring chemicals that are released by bacteria, in this case, that prevent the growth of other bacteria. And he won the Nobel Prize in 1952 for the discovery of streptomycin, which is effective against tuberculosis. So these chemicals, some of them are purely chemicals and some of them are compounds that are produced by live cells, but these are both all antimicrobial agents. But circle Alexander Fleming, that's the guy you really, really want to focus on. But there is a history there, just like anything. Now, here's a picture. This is not his actual slide or his actual agar plate, but you can see here an agar plate where they have done the technique of streaking Staph aureus, which is the bacterium, on the plate using the streak for isolation method. You probably recognize streak in zone 1, then flame the loop, streak in zone 2, flame the loop, and then streak in zone 3, and you start to see the individual colonies. And then they have this colony, maybe 2 colonies of a fungus growing on the plate. And this is penicillium. So this could be similar to what Fleming saw on his plate. And what you notice, though, is right around the penicillium fungus colony, you have this area where there's no growth of the staph. And so he noticed this and he said there must be something being released from the penicillium into the agar, and then it diffuses away from the colony. And at high concentrations, meaning closer to the fungus where the penicillium is releasing the penicillin, then the staph cannot grow. But as the diffusion occurs in the agar away from that fungus, eventually the level of penicillin in the agar becomes weaker and weaker, and there is a point at which the staph can grow. And so the zone of inhibition is the point of where the penicillin chemical is at a high enough concentration to prevent the staph from being able to grow. Now, penicillin is a natural product of a fungus. They took penicillin, though, scientists took penicillin and chemically altered it to make some drugs we call the semi-synthetics of penicillin. So they made some chemical alterations that make them a little bit easier to use or easier to store, or usually that's it, easier to store because they go bad on the shelf if sometimes when they're in their natural form. So they chemically alter them to make them longer lasting or more effective. And the semi-synthetic that you might be familiar with is called methicillin. So there are some other ones too, but penicillin is a natural product of the penicillium fungus, and then scientists chemically altered the penicillin to make methicillin, which also inhibits bacterial growth. You may also see some antimicrobials that are completely synthesized in the laboratory. they're synthesized from scratch from chemicals in the laboratory. Those would be called true synthetics, meaning they've been synthesized from scratch. But semi-synthetics means you take a chemical that naturally occurs and you just change it a little bit to make it a little better. So we have a lot of natural antibiotics and semi-synthetic antibiotics that are available for doctors to use when a patient has an infection. And there's certainly some full synthetics. I'm going to get rid of my picture. I don't think you need to see me. So on this slide, I'm giving you a list. You do not need to memorize the list, although on the right side, there's a few that we are going to focus on. But on the left side, you see this list. And the list shows the scientific names of a couple of fungal species, and then a long list of bacterial species. And there are drugs, natural and semi-synthetics, that have been extracted from these species. So penicillin, and then some other types of antimicrobials from fungus, but penicillin, that's one you're definitely going to need to know. And then you may recognize something on the list of bacterial natural antibiotics. So I'm highlighting bacitracin, polymyxin. You may have some familiarity with some of these other ones, streptomycin, neomycin, tetracycline, chloramphenicol, amphotericin, anyway, ivermectin. So you may have some familiarity with these. I'm going to just pick some, but that we're going to focus on a little bit more. So penicillin is one that you need to focus on. And then over here on the right, bacitracin, polymyxin, and neomycin. And the reason I picked bacitracin, polymyxin, and neomycin is they're particularly in products that you may recognize. So I put Neosporin here. When you're in the drugstore, if you start to look at some of the ointments and such, you'll recognize some of these antibiotics that are in them. So penicillin, but you don't need to know the names of the bacteria that make bacitracin, polymyxin, or neomycin. So, but there is a bacterial species that naturally produces each of these molecules. So what are antimicrobials? They are, when they're used in the body, what's important about them is that they have selective toxicity. That's a concept that we've discussed in the past, meaning you want to kill the microbes, but not the patient. So any chemotherapy needs to kill the microbe, but not kill the patient. The antibacterial drugs are the most common of the antimicrobial agents. Antifungal drugs and antiprotease drugs are much less available. There aren't as many drugs that we have been able to find to treat eukaryotic cell infections. So if a human is infected with a fungus, it's harder to, there isn't a large selection of drugs that are antifungal. There aren't as many different drugs that are antiprotease. But there are actually quite a lot of drugs that are antibacterial. So that's what I'm trying to say here. There's also a pretty limited amount of antivirals. So in the next lecture segment, we will talk about the different mechanisms of action that microbial drugs in the body have. And you'll notice we're focusing mainly on drugs that affect bacteria, because in this image, you have a bacteria in the center of the image here. And then a few antivirals and a few antifungals.